Hi student, in this video you are going to learn about the chemical digestion of amylose and amylopectin. Before I start explaining the way amylose and amylopectin are digested chemically, I would like to remind you about the basic structure of alpha D glucose. If you remember your chapter 2 topic, you have learned that glucose is a 6 carbon ring molecule. Each of the carbon is numbered by 1, 2, 3, 4, 5, and 6. The first carbon is the carbon that is next to the oxygen that has no branches, while the last carbon, which is the sixth carbon, is located as the branch of the C5 or carbon number 5. So you can see from this structure, the first carbon or C1 is located next or to the right side of the oxygen, and then C2 is the next to it, go and on until C5, and there is a branch that has the C number 6. Glucose is a monomer, which means that they can be combined to create polymers. Now, in picture 1 of this slide, you can see two alpha D-glucose that are combined by this blue linkage or bond. This happens through the condensation reaction. I hope you still remember the condensation reaction, in which that the OH or the hydroxyl of the first glucose and the hydrogen of the second glucose will be combined to become water. So water is the side product of the condensation reaction. After this water molecule is withdrawn from the two molecules, then there will be a new bonding, which is called glycosidic link in between the two glucose, which has an oxygen in the middle. Now that's what you can see as the representative of this blue bond. Okay, so picture number 1, the blue bond is described by the condensation reaction in the picture below it. Now, in picture number 1, you also can see the yellow line. This yellow line is also a glycosidic link, but the difference is rather than between carbon number 1 and number 4, this glycosidic happens between carbon number 1 and carbon number 6. All right? So this will create a branch. Now, if one big molecule, the polymer of the glucose, has only one type of the link, which is 1.4 link, the blue one, it will create amylose. As you can see in this table, okay, next to picture 1, amylose has only one type of bonding, which is 1.4 glycosidic bond. And it will create a linear molecule without any branches. On the other hand, you can see amylopectin next to the amylose. Other than 1.4 glycosidic bond, it also has 1.6 glycosidic bond. Because of the presence of 1.6 glycosidic bond, you can see that this molecule is no longer one straight chain, but it has lots of branches. Now, remember, 1.6 glycosidic bond only happens one for each branch. The branch will get longer by adding more glucose as a straight line, which means that it is 1.4 glycosidic bond that is used to create a longer branch. But the main point or the first point of the branching happens because of 1.6 glycosidic bond. I hope you still remember also from your chapter 2, enzymes are specific. So you can create an analogy like key and lock model. So a specific key can only open a specific lock. The same thing also happens here. You have learned in the previous subchapter that to digest starch, you need amylase. Amylase will cut the starch into maltose. So you can see from this picture, imagine this is amylase, a long chain of glucose. Maltose will cut this amylase into maltose, okay, disaccharides. Therefore, amylase cannot fully digest amylopectin. Why? Because amylase can only cut the blue lines. Okay, every 2 glucose, the blue line or the 1.4 glycosidic bond can be broken by amylase. However, the yellow bond, which is 1.6 glycosidic bond, cannot be cut by the amylase. So if we try to cut every 2 glucose in this molecule, the amylopectin molecule, you will find that at the end, there will be this molecule called dextrin that cannot be cut further by the amylase. So the definition of dextrin here is the fragment of amylopectin molecule containing 1.6 bond or glycosidic bond that cannot be further cut or digested chemically by the amylase. Therefore, there is another enzyme needed to do or to complete the process of chemical digestion of starch, specifically amylopectin. The enzyme is called dextrinase. It cuts 1.6 linkage or bond between the alpha D-glucose. So you can see from here, as soon as there is dextrinase, the yellow bond represent 1.6 glycosidic bond is cut in between these 2 maltose. So you can see now all the molecules from the amylopectin have become maltose molecules. To complete the digestion of both amylopectin and amylose that are now in form of maltose, there is another enzyme that is required to complete the process. It's called maltase. Maybe you can guess that maltase, from this picture, you can see it will cut the maltose into individual glucose. Now the glucose are ready to be absorbed by the villus, or the, in plural, it's called villi, of the small intestine. That's the journey of chemical digestion of starch that consists of amylopectin and amylose. And this is just for your information. If you want to know the content of amylose and amylopectin in rice, you can see from the first picture on the left, regular steamed rice that we usually have every day has low amylopectin content, while the sticky rice is rich in amylopectin, and it is the stickiness of the sticky rice that is caused by the amylopectin. Because when amylopectin is reacted with the hot water, it will become gel like liquid, so it becomes something sticky. So for your information only, so you can see how amylose and amylopectin exist in the rice, and you can detect their existence.